Hello, everyone, and welcome to today's webinar series and this episode of Discover Molecular Interactions. Each month, we are looking at a different molecular interaction and we discuss its challenges and also present some essays how to analyze them. My name is Vivian. I am responsible for scientific communication at Dynamic Biosensors, and it's my great pleasure to present to you our guest speaker of today's webinar episode, Dr. Marc-André Kasper from Tubulis. Hi, Marc. It's great to have you here.
Hi, great to be here.
So Marc is the Vice President for Chemistry and Early Discovery at Tubulis. And in today's webinar episode, I will just start with a very brief introduction telling you how you can measure real-time on and off rates directly on cells. And then I will directly hand over to our guest speaker today. And Marc will tell you all about Tubulis' platform for antibody drug conjugate development and how they used RTIC in their pipeline. If you have any questions during the webinar, please feel free to use the Q&A feed or also use the normal chat function, and we will answer all the questions in the Q&A session after the talks. For those of you who have never met Dynamic Biosensors, we are focusing on kinetic measurements. So we look at two interactants that interact with each other, and the interaction is usually described by the association rate K on and the dissociation rate in the unbinding event K off, and also the resulting affinity Kd. So if we look at the next slide, the reason why we are looking at cells is, as you can see, the cell membrane is much more complex than if you look at an isolated ligand-analyte interaction. So you have all the co-interactions, you have the density of the proteins within the membrane, the proteins can move around in the membrane. So it's very important to look at interactions directly in this complex native environment. So this is why we are measuring this with real-time interaction cytometry, and how we do this is described on the next slide. So it is a real-time measurement, and this is important because the on and off rates always give you more information than just simple affinity. So there are also other technologies like flow cytometry, for example, where you can do titration-based measurements and you will get a KD out of that. But if you look at these two examples, these are two very different kinetic behaviors. The left example is very fast, whereas the right example is rather slow, but still both have the same KD. So that is why we think it's very important that you measure the real-time waves, as they will give you the whole picture of the kinetic measurement. And our instrument on the next slide is shown here. It's a chip-based method. So you see the chips in this image, and on the chips we have these tiny cell traps. These are polymer cages which are biocompatible, and they allow us to trap single cells, living or fixed, adherent or suspension cells, on the surface of a chip. And on the next slide, you can see the typical workflow. So we have a microfluidic channel on the chip. The cells are flushed in, they get stuck in these traps. You don't need to culture them on the chip or have any anchoring molecules. And afterwards, you need a fluorescently labeled analyte molecule. And when this analyte molecule then binds to the receptor on the cell surface, you will see an increase in the fluorescence signal. And from this, you can then derive the real-time association rate K on. Then instead of the analyte solution, we use simple buffer, and this will then allow the analytes to dissociate again, and you will see the decrease in fluorescence. And from this, you can see you can derive the off rate. Afterwards, you can also apply a reverse flow, just flow the buffer in the reverse direction to push out the cell again, and then the chip can be reused. So this is the basic principle, and now I don't really want to use more of the time, and we'll hand over to Marc, and I'm very excited to hear about the application example.
Yes, thanks so much for this kind introduction. Also, thank you for having me today for this webinar series. Today, I would like to talk about how we leverage topoisomerase 1 inhibitors for the treatment of cancers with a very interesting modality, which I would like to explain in a second, which we use, which is antibody drug conjugates. And I would also like to discuss how we use the technologies that were just introduced to biophysically characterize our molecules to be ready to apply them in the patient and get all the sufficient data. But maybe first of all, for those of you who have never heard about Tubulis, a couple of words to the company. We are a Munich-based biotech company, currently 45 professionals working for us. We are developing antibody drug conjugates. As I said, I will talk about this modality in a second. Actually, we have 2 solid tumor targeting assets, which are just about to enter the clinic. So we are just making the step to be a clinical stage biotech company very soon. Next to these assets, where we are addressing 14 out of the 15 most fatal tumor types, we are also still innovating on all aspects on the ADC. In particular, my team is focused on unlocking novel payloads to be delivered with ADCs. Furthermore, we are leveraging selected collaborations. For example, we were able to get a licensing deal with Bristol Myers Squibb earlier last year on a limited number of ADC programs. And we can do what we can do finally because we are backed by a strong investor base with European and US-based investors, where we recently closed our Series B2, which helps us to do what we are doing and to bring benefit to the patients. But first of all, a couple of steps back. So let's start with our motivation. Our motivation is the growing cancer burden worldwide. I don't have to tell too much about this. Usually, someone knows. Everyone knows someone who gets diagnosed with cancer in their close surroundings. So currently, or into 2020, there were 19.3 million cases and almost 10 million deaths globally. And this is expected to even increase to 28.4 billion cases in 2040. So it's a pretty deadly disease, as we all know. So let's have a look what we can actually do about this. And the most commonly applied treatment against cancer is still chemotherapy. This is usually the first line the patient receives. So chemotherapy is designed in a way that it's given to a patient systemically. It can divide into the whole body, and it mainly acts on rapidly dividing cells, as the tumor is. But of course, also other cells are rapidly dividing, which also will get harm from this chemotherapy. So it's very effective in eradicating cells, but it's not so much specific for the cancer. So therefore, people came up with more targeted approaches. So with targeted therapy, for example, with monoclonal antibodies. These are these huge Y-shaped molecules that you will now see throughout the presentation. Monoclonal antibodies can be designed in a way that they specifically bind the tumor cells and spare healthy tissue. And by this, they already can have an effect on the tumor side, for example, by getting a recognition from the immune system, or by blocking growth factors to bind, and thereby have an inhibition on the tumor. However, they can lack efficacy in the patient because these modes of actions are not so efficacious as chemotherapy, for example. So it's very specific, but it's not that effective in the end. And therefore, people came up with a very smart approach, as we think, which is targeted drug delivery, which basically takes the best out of the two worlds. We have the specificity of the antibody for the tumor and the highly effectiveness of the small molecule chemotherapy. And we use the antibody to guide the chemotherapy to the patient in the tumor. And this is what we are also doing. And we are not the first ones who invented this principle. So actually, the first approval of an ADC was back in 2000, so more than 20 years ago. Until then, 12 ADC made it into the market, into the clinic, with a very large pace in the last 5 years, as we can see here. However, there's still a lot of room for improvement when we look into the ADCs that are currently being used in the clinic. And one of the issues, it's not the only, but one of the issues which is often discussed, is instability when these ADCs are circulating in the patient. So usually 9 out of 12 of these ADCs, which you can see here, are linked via a chemistry which is called maleimide chemistry, and this can undergo a process which is called retro-Michael addition. And this leads to a loss of the cytotoxin or of the chemotherapy in the patient while circulating. So the actual picture that we have more looks like this, where some of the cytotoxin gets transferred to the tumor by current ADCs, but not all of it. And this, of course, leads to less efficacy than we could expect. And it can also, of course, lead to off-target toxicity. But this is not the only problem of current ADCs. There's other things. There's unspecific uptake of the whole ADC into tissue which is healthy. There's also aggregation. So it's not only it's not the only answer to make things stable. It could actually making things stable could actually make things worse because it could trigger aggregation. So this is what people tried. So it's a bit more complex than people thought in the beginning, and you really have to innovate on many aspects of the ADC to actually make a good molecule in the end. And this is where Tubulus comes into the play. So we have many technologies, or a whole toolbox of technologies available to actually make good ADCs and to innovate on all aspects. We have something we call the TAPTEC conjugation, where we can make homogeneous DAR2 and DAR4 ADCs, which means that we can directly and on a specific place place 2 or 4 drug molecules or on one antibody. We can use something that we call the P5 conjugation to make homogeneous DAR8 conjugates. I will talk about this in a second. And we are also interested in the payload. So what do we actually deliver to kill the cancer cell in the end? And here we have a strong focus on topoisomerase 1 inhibitors, but we also want to look beyond this. And this is especially the focus of my team, think about new linker payloads. But let's, for today, focus on P5 in combination with tubutican, or with topoisomerase 1 payloads. This is how we call them. So first of all, what is P5? So what can we do better than others? So we need to somehow connect the drug to the antibody. Again, the antibody is this large Y-shaped molecule, and we can use this molecule in its native form and leverage the disulfide bonds. Usually an IgG1 format has 4 disulfide bonds, which we can selectively reduce to liberate 8 cysteine residues, and we can now do an alkylation with the drug and thereby make the connection between the drug and the antibody. And usually here are people, or the ADCs on the market are using maleimide technology, and instead of this, we use something that we developed, which is called ethynyl phosphoramidate modification, to actually make an ADC, which is superior over what is currently in the clinic. And it's superior because we can easily incorporate this. I will show you this in a second. We have a very simple labeling process. We do not need much access. Most importantly, we have a very high linkage stability. I will also have some data on that. And we have, for the chemist in the audience, we have another substituent over here at the phosphorus, which we can use to incorporate hydrophilicity and thereby also conjugate very hydrophobic drugs. So how does the labeling process look like? As I said, we're starting off from native, usually IgG1 antibodies. We just incubate them with our linker payload, carrying the P5 motif and TSEP for reduction. It's a one-pot protocol. It's very fast. And because it's so fast, we usually do not need a large access. So when we use 10 equivalents of the labeling agent, we end up with 8 molecules on the ADC. So per cysteine, we only need 1.2 or 1.25 equivalents. So it's very efficient. And most importantly, since we have 4 disulfide bonds to liberate 8 cysteines, which we fully alkylate, we end up with a homogeneous molecule, which is only DAR8, so only 8 molecules on one antibody. And this we can see in the analytics. So we have a very clear site exclusion profile. We also have a very clean, only 1 peak hydrophobic interaction chromatography profile. In the MS, we also usually only see DAR 8.0. So it's very simple. We are also able to show that this is scalable, and we scaled this now to a kick, so a kilogram of ADC output without any larger issues. But of course, there's one important thing which we do not want to lose. So when we think about our antibody, this is our targeting molecule. So our antibody needs to find the tumor cell and needs to deliver the cytotoxin. So what we do not want to lose is the affinity of the antibody while conjugating the drug for the targeted cell. And here's where we teamed up with dynamic biosensors with this fantastic method where we can really measure the binding kinetics of the antibody and compare it with the ADC that we actually made. And the outcome you can see here. So we compared here different molecules. We used the unconjugated antibody, which is an HER2 binding trastuzumab in this case. We used an ADC that we made with our technology out of this. And we bought an ADC from a pharmacy, which is called Enhertu, which is also based on the same trastuzumab antibody. And we measured these, or dynamic biosensors measured these constructs for us with RTIC. And here you can see the outcome for the trastuzumab. So it has very nice binding kinetics, as we would expect it from this antibody. It's a therapeutically approved antibody, so it's with a good kinetic, good K on and K off rates. Dynamic Biosensors was also able to show that this works for living as well as for fixed cells with this construct, and the results in the end were very similar in terms of K off and K on, and also for the KD. So we had a look and wanted to know how the tubulin ADC, so after the conjugation, now behaves, and we were very happy that this looks very similar. It also works on living and on fixed cells. This was clear now. We see a slight reduction in on rate, but still the KD, when we look at it, it's very similar to the trastuzumab. So we are still in the picomolar range and below 1 nanomolar. We also did this for Enhertu, and it's also similar. It's this time we only looked at this at fixed cells, and when we now compare all these 3 conjugates, we see that the conjugation has some influence, but it's very minor on the binding affinity of the antibody. And even though we compare ourselves with a material which could go into the clinic, we are still having a better KD compared to Enhertu with a material that we generated in our labs at Tubulis. So we were very happy to, or very pleased to see this data, and we could take this data with a lot of confidence because we knew it was measured not on an ELISA setting with isolated protein or so, but really in the native environment of the cell. So we moved on with our data and wanted to see what is actually the outcome of this retained binding of the properties of the ADC. And what we see here is cell killing of HER2 positive cancer cells. And we compared, again, our technology, so trastuzumab tubulin, with something we bought from the pharmacy with Enhertu. Again, same antibody, very similar payload, but our linker. And we were very pleased to see that the binding obviously is still there. So on HER2 positive cells, we see a very good dose-dependent effect. So these cancer cells die at higher concentrations. We even see a better effect with trastuzumab tubulin compared to Enhertu, especially on cells where HER2 is not that overexpressed, which we're very happy about. And we do not see much of an effect on HER2 negative cells. So it directly shows that the binding that we measured before also holds true in other readouts. Of course, we wanted to know, does it really hold true that our conjugation platform is that stable? And when you remind my introduction, I showed you that approved ADCs such as Enhertu have this problem of instability, and this is what we can see here. So we took Enhertu again from a pharmacy, incubated this in rat serum, and you can drastically see how this loses its payload. So in the very beginning at day 0, it's 8 payloads per antibodies. This is how it comes. And even after 1 day, this reduces almost by 50%. And after 3 days, there's only 3 molecules per antibody in an average left. And here you can see how it's meant and how it looks like for us. So we are fully stable over the whole observation period, and every payload stays on the antibody. So this is the increased stability that we see by using the P5 conjugation platform. We also looked into this in vivo, and this is now a similar experiment. We just injected mice with the 2 substances and checked also the drug-to-antibody ratio via mass spec over time. And here we also see that Enhertu really also in an in vivo setting loses its payload while we are fully stable. So Enhertu, if you remind, or if we go back to our human scheme here, rather looks like this, that the toxin doesn't really get transported to the site of action, but it's lost during the circulation, where the tubulin ADC directly brings the payload where it belongs to the tumor. We also saw that we in general have a very good PK behavior of these ADCs. So you could imagine if you put 8 hydrophobic molecules on an antibody, or what is known in the literature, this can be challenging in terms of pharmacokinetics. So sometimes these ADCs, especially with these high loadings here, rapidly clear from circulation because they do have hydrophobic interactions and things like this. But here we also have the big benefit that we have these hydrophilic moieties that we can see as an R, which really gives us antibody-like pharmacokinetics. So here we injected rats and just quantified either the, so we injected rats either with the unconjugated antibody in green or with the ADC, and we see the pharmacokinetic profile. So the clearance from the rats in vivo is very, very similar. So we do not change the properties of the great properties of an antibody via the conjugation. But there was another question that came up. You know, the more that you think about your molecules and the more experiments you do, the more questions also arise. And at one point we had the question, do we actually change the tumor penetration? If you remember, we want to treat patients with solid tumor in the end. So our molecules also have to diffuse into the solid tumor. And what is known from the literature also is that the bigger a molecule is, the less tumor penetration is usually possible. And we wanted to find out, I mean, the linker payload is not large, but still we increase the size of the antibody by conjugating the payload. Does this have an influence on tumor penetration, or could this potentially be? And for this, we wanted to find out how much is this conjugation that we are doing when we are turning an antibody into an ADC, influencing the hydrodynamic radius of the molecule, because the hydrodynamic radius is what is important when we think about tumor penetration. And here we again teamed up with dynamic biosensors. This time we used the helix instrument, and we did again a comparison where we wanted to compare the hydrodynamic radius of an unconjugated IgG1 with our ADC, again with Enhertu from the pharmacy, and we wanted to have something which is known, it's too big to penetrate solid tumors, and this is also known from the literature. These are these large IgM molecules, which we also used. And we used the, as I said, the helix instrument in the dynamic mode. And I'm sure people from dynamic biosensors can explain this much better than I do, but I will try my best. So what happens here is there's a currency applied, and thereby the analyte is taken to the surface, and there's a fluorescence reporter here, which gets quenched when it's close to the surface, and afterwards it just goes back in its original stage, and the larger the dynamic radius is, the longer it takes until the analyte comes back here. And this is thereby a very good measurement to see actually how large the molecule is in terms of hydrodynamic radius in aqueous solvents. And the value is, so here we see this reflux, so this after the molecules were taken to the surface and they relax, the longer it takes, the larger the hydrodynamic radius, and there's something that was defined by dynamic biosensors, which they call a lag value, and this is the area above the curve. So it just means what we need to know for the next slide: the larger the lag value, the higher the hydrodynamic radius in the end, and this is something we do not want to increase. And here's the results that we generated with this. So when we look into the lag value of an unconjugated IgG, we are very pleased to see that the two Boutiquin ADC is basically the same. So even though we increase them in size, we do not increase them in radius. We are even a bit lower in radius than Enhertu. And this was a bit surprising since the molecular weight of Enhertu is a bit smaller than what we actually add to the ADC, but we were very happy to see that. And of course, we were far away from our control, from our IgM. So this was a very good way for us to get more confidence that we can actually deeply penetrate the tumor with our ADC, because we do not increase it a lot in size. And also here we did a nice confirmation experiment, and this is now an in vivo model where we treated tumor-bearing mice either with our trastuzumab boutiquin-based ADC or Enhertu. And here we really showed that we have a very good tumor penetration, because over all dose levels, so from 2, 1, 0.5, and 0.25, we were superior in eradicating the tumors from these mice. So Enhertu is shown here in gray, we are in orange, and I think most exciting is when we just look into the doses at 1 mg per kg, so both ADCs at 1 mg per kg. And here we see that our technology really has a feature to have a long-lasting effect on these tumors, whereas Enhertu is quickly growing out after a short efficacy period, which we also know, I mean, it's an approved medication, but our technology really enables these long-lasting effects and these long-lasting eradication of these tumor cells here. And here I already come to my summary. So today we talked about a cysteine-based antibody conjugation platform, which we call P5 labeling. It enables us to generate homogeneous ADCs in a very quick way, in a one-pot reduction and conjugation process. We only need minor excess of the cytotoxic material. This is, of course, important because it's expensive and also toxic. We have combined this with a topoisomerase 1 linker payload platform, which we call tubotecans, which has excellent biophysical properties and very strong in vitro activity. With the help of dynamic biosensors and the helix cyto and the helix, we were able to show that we have a retained antibody affinity to its target, even though we modify it. We do not have any influence on the hydrodynamic radius after the MAP conjugation, which is good for tumor penetration. And our platforms, our tubotecan platform enables us to get highly loaded DAR8 conjugates, with a very good in vivo efficacy, directly compared to an improved medication based on the same antibody and a very similar payload. And since we generated all this nice data around this platform, of course, we want to use it. We want to bring it to a patient. I mentioned this in the beginning. And we're using this for our program. So our pipeline, you can see here, our two front runners are TAP30 and TAP40, targeting 5T4 and NaPi2b. These will, as I said, soon enter the clinic, TAP40 a little bit earlier. But of course, this is not it. We are also interested in novel targets, which are yet undisclosed, which bring even more innovation to the field. And we are also using this platform with our partners. So we were also able to convince Bristol Myers Squibb to work with this platform on a selected number of targets, which they are also now proceeding to clinical studies. And with this, I'm at the very end of my talk. I thank you all for your attention. I also thank all the people at Tubulis, in particular the people who are highlighted in orange here. These are the people who are doing the heavy lifting in the lab all day and help us to generate the nice data that we were just able to see. And of course, well, thanks a lot to Dynamic Biosensor for having me today. Now I'm ready to take questions.
Thank you very much for the great talk. It's really amazing to see how this promising strategy is now making its way to the clinic. Yeah, we are opening the Q&A session now, so please feel free to write anything you want in the Q&A tab or also in the normal chat, or you can also talk to us directly. If you raise your virtual hand, I can unmute you and we can chat. And maybe I just start with a first question. So from an early discovery point of view, so what do you think comes next now that this goes into the clinical phase? What would be the next point you would like to focus on? Is it going more into developing other antibody formats like bispecific formats, or are you more interested in further discovering other linker properties, or what's next for you there?
So what we are especially my team is most interested in finding new payloads, because I mean, what we have seen is all the ADCs that are in the clinic at the moment, these are 12, and they just rely on 3 different mode of actions how to kill the cell. And these are basically killers for these cells. So it's either tubulin interaction or they interact with the DNA, and they are killers for all cells. And we are trying to make now the drug also a little bit smarter and maybe selective for the cancer cell, because in that case, we could even improve the safety of these molecules beyond just improving the linker, but also, for example, when you think about the whole ADC gets taken up by a different cell, we could also eradicate a risk that comes from this by making the drug smarter. And this, I think, is the next innovation step in the field.
So more other chemistries for the drug targeting, yeah. That's true. That would probably also help other fields.
But probably this is the case because you're talking through the chemist in the company. Maybe our biologists would say something differently.
Well, okay, I don't see any questions yet. But there are in the Q&A section. So there are some technical questions about the method. So maybe I will answer them directly. The first question is whether the method is compatible with small molecules. Yeah, you can do RTIC with small molecules. The only requirement is that it is fluorescently labeled. So your analyte molecule always needs to be fluorescent for us to be able to detect it. So either you have a method to directly label your small molecule, or if that is difficult, you could also use indirect approaches like a competition assay and have another labeled competing component and then have your small molecule unlabeled. That would be the two options there. Okay, but the second technical question I don't see so far, but there's a question in the chat. That is a question for you, Mark. Is there a particular reason why the conjugation was Malay-Mead based, whereas NHS EDC chemistry could also mediate antibody conjugation?
Yes. So the problem with the NHS chemistry is that it's relying on lysine residues for modification on the antibody. And the problem is that there are almost 100 lysine residues on one antibody, but you do not want to place 100 drug molecules on an antibody because it would just precipitate, because these drug molecules are usually quite hydrophobic. So what you usually aim at is something like a DAR2 to a DAR8 format. And if you now think you want to modify these almost 100 lysine residues only with 8 drug molecules, you end up with a very heterogeneous mixture. So potentially you could have a couple of million different ADCs then, and it would not be defined. And this is something that regulatory authorities usually do not like too much. Therefore, people like to use cysteines because you have these interchain disulfide bonds, which are in an IgG1 format at least very defined, and you can liberate 8 conjugation sites, which you can then react with a maleimide, or we think even better with an ethynyl phospholamidate.
And could you comment on why your conjugation strategy is superior to the maleimide chemistry? Because you showed the serum stability and your antibodies are very stable, but you are disrupting the native disulfide bonds. So
Yes.
why is it still so stable?
The native disulfide bonds are actually not necessary for the stability of the antibodies themselves. So they do not fall apart even though you remove them. And the reason for this is that the non-covalent interaction between the heavy chains and the heavy chain and the light chain is so strong that it compensates for the loss of the covalent disulfide bond that you remove. So you do not see them falling apart. And the stability really comes from the chemical stability in the linker. Since we are not relying on maleimides, so in fact, this is not a Michael system, and therefore we do not have the reverse addition and chemically cannot lose it. That's a good question. One could think that if you remove the covalent bonds between the chains, they would fall apart. But yeah, as I said, since the non-covalent interaction can compensate for it very nicely, it's not an issue.
And why do you think that this does not affect free cysteine conjugation from the variable domain and also not the eventual binding of the antibody?
Yeah, usually we make sure when we select our antibodies that we do not have cysteines in the variable region, and they are also rarely found. And usually from a development perspective, you would also would like to have them in, since they cause problems during expression on the antibody on a large clinical scale already, because they do form disulfide bonds with each other, and thereby, precipitate, aggregate, and do all these nasty things that you do not want. So usually a therapeutic antibody doesn't have a cysteine anywhere else than trapped in disulfide bonds.
Now I will quickly switch back to the technical question on the RTIC method, whether one can use indirect labeling. So as I said, you can use a competition assay, but this was more with regard to, for example, streptavidin and biotin systems. So you can, of course, also try to use a streptavidin and biotin system. The only thing you always need to make sure is that the streptavidin is really stably bound so that you know what are you looking at. Are you actually looking at your small molecule dissociating, or are you looking at the streptavidin biotin dissociating? So yeah, but I'm also happy to discuss more detailed specific projects later on. So I will later on write you my email address, and then we can reach out and talk about this a bit more specific afterwards. And there's also a question about internalization, whether we saw any internalization during the measurements of these ADCs. Internalization is something we are very strongly looking into it at the moment. Of course, it's a bit difficult to tell because the fluorescence will still be detectable if it's in the cell, whether it's outside of the cell or in the cell. So we cannot really differentiate whether something is just binding very strongly or whether it's internalized. So I can't really tell you much about this for now, but we are working on dual color assays at the moment. So you can measure in two colors. So then you can play around with like pH sensitive dyes to really differentiate between internalized antibodies versus bound antibodies to the surface. So that's definitely something that's very interesting for us too. Then there's another question about the linkers, whether you have used enzyme-activated linkers in the ADCs, or if you have any suggestions in choosing this type of linkers.
Yes, we do. So with enzyme activation, I think you're referring to a cleavable linker, which releases the payload after the ADC is taken up into the cell. And yes, we do. And also we do think it's crucial for a couple of reasons. First of all, it's dependent on the payload that this gets really liberated, for example, by a protease to have its full efficacy inside of the cell, because yeah, when you do not tracelessly release the payload, then sometimes it's not that active anymore. But even more importantly, it's important to have something like a bystander effect, which only works when you release the free payload in the cell, which helps to eradicate heterogeneous tumors where not all of your cells in your tumor actually express the target. So long story short, yes, we use protease activatable linkers to liberate the payload, and we think it's important to do so.
Okay, if you have any further questions or some more specific questions or something comes up later, please also feel free to reach out either to Tubulis. I'm sure you have your contact details on the website, or also to Dynamic Biosensors. Okay, I hope I didn't miss any question. If I did so, I'm make sure to get back to you after the session. I will write our email address in the chat, so you can reach out whenever you have any other questions. And please feel free to follow us on social media and check out our website. We have other webinar episodes coming up soon, and also previous episode recordings that you can find on YouTube. So I would be happy to see many of you in any future episodes. And thank you so much, Mark, for this nice presentation and for showing us the data and your ADC platform.
Thank you for having me. It was a pleasure.
And with this, thank you all for joining us today, and I wish you a great rest of your day. Goodbye.
Thank you. Bye-bye.